Hello, everyone. My name is Michael Schatz, and I'm your host this week for the Galaxy Training Academy's Meet the Experts series. The goal of this series is to hear firsthand from leading experts from around the world on some of the most important topics in the use of Galaxy, computational biology, data science, and beyond. Throughout this week, we'll release one video a day to do a deep dive into human genetics, single-cell and spatial transcriptome analysis, proteomics, conservation and genomics, genome assembly, comparative genomics, and more. Our guest today is Dr. Delphine Larivière. Delphine is a bioinformatician and genomics researcher at Penn State, where she works in the lab of Anton Neradchenko. Her research focuses on developing computational methods and workflows for analyzing large-scale genomic data, with particular emphasis on reproducibility and accessibility for genome assembly. Notably, she develops the assembly and annotation workflows for the Vertebrate Genomes Project. Delphine is also an active contributor to the Galaxy Project, helping to build tools and developing training resources that empower scientists to perform complex analysis on a variety of data science topics. To learn more about her, you can find her online in the Galaxy Element channels and as Delphine-L on github.com. Delphine, thank you so much for joining us today.
Thank you for having me.
All right, great to have you today. So what I'd like to do, first we talk a little bit about you and your background, then we'll talk about the Galaxy and the Training Academy, and then I hope at the end we can talk quite a bit about the VGP and your sort of work that you're doing there. So first of all, how did you get started? Were you always interested in genomics?
I was always in between like math and biology when I was in high school, and I didn't learn really about bioinformatics and biostatistics until later in college. And when I discovered informatics, I'm like, yeah, that's my jam. It has
Yeah.
like both worlds. And yeah, always, I think when I was a kid, I wanted to be like a virologist for some reason. So I'm not too far off. I worked on some virus at some point, but yeah.
What was your interest there with viruses? Where did that come from?
Uh, I'm not sure. I was a bit of a goth kid, so everything that was like dark and disease
Ah.
and like, but also still like biology and science. I was kind of a middle ground, I guess.
Awesome, awesome. And then how did you end up at Penn State?
So I did my PhD in CIRAD in France, and we had a Galaxy instance there that we used to like share our tools with the users. And I really enjoyed, found that like a great tool to collaborate and to work with. And when I finished my PhD, I'm like, yeah, I mean, looks like you guys have a great community and you were hiring. I'm like, okay, and that was 10 years ago. So
Oh, excellent, excellent, excellent. I don't know. I mean, I think you said you were using Galaxy as a PhD student as a way to kind of, I don't know, distribute tools and kind of show off your work. Were there other kind of parts of Galaxy that were of interest to you then?
um, I think that was, I'm not sure what you mean, but like, yeah, like we had a Galaxy instance, and we were using it, like we were maintaining it for the whole
Yeah,
institute. So that's how I got to know it. But I saw it was like a whole, not just a tool, but also like a community of scientists. Like
yeah, yeah,
there was
yeah.
movement there. It was not just like a team of like developers and there was like a lot of scientists involved in it, and it was good atmosphere, I think.
Excellent, excellent. So now that you're at Penn State, what sort of roles do you have now?
Rest of the roles.
Yeah,
Um, so I don't have like this is what I like a lot in this position is that I get to collaborate with a lot of different projects.
yeah,
So my role is to connect with people who have data or have a problematic, and to help them identify what to do with the data, what are the best tool to use, and put everything in Galaxy and spread it to the whole community, both for them to share their work, but also so that the work we do together is not lost and it's available for everyone. So that involves like going through understanding the process, finding the tools, writing the training, and giving training and communicating about it. So it's super varied. I worked on bacteria, viruses. Now I work on vertebrates.
yeah.
Um, so really love it.
Awesome, awesome. Yeah, I mean, you brought up training a few times. Maybe you could say a little bit about like what is the Galaxy Training Network and what is the Galaxy Training Academy? What are those all about?
The Galaxy Training Network is a platform that is a huge resources for both the Galaxy community, but also outside of it, because we have a lot of training on a lot of different topics. We go from climate to proteomics to genomics to just basic Python. And you can learn at any level you are. You can either learn like, oh, I'm discovering this topic. Where do I start? Also like, oh, I'm kind of familiar. What are the latest tools? What are the latest things that people are doing? And there's a huge community both using it and maintaining it. And I find it very interesting because it's such an inspiration for me to see how people are involved in sharing their knowledge and sharing um, learning things. And the GTA came from, okay, we have this whole resources, bunch of resources, we're all giving like local training, but how can we make it available to everyone? Because we have a strong core team in the US, we have a strong core team in
Yeah.
Australia and Europe, but if you're not like close by to this people, you don't really get access to like training by us
Yeah.
directly.
Yeah.
So
Yeah.
let's do that online. And it started with the smorgasbord um, long time ago. I don't remember when the first smorgasbord was, which was a gigantic event. But it was a lot of work. And I think it was happening every other year. And so we changed the formula with the GTA, which is a bit more reasonable, a bit more manageable, and we can have it every year. It's still so it's an online event. Everything is asynchronous through video. The video are available forever once we created them. But it's free. Everyone can participate. We have more than 1,000, maybe 2,000 registration every year, and that's all year is doing that. And it's great to see that we can reach people who are from Africa, from Asia, from like far away from where our teams are. Uh,
Yeah.
and we can get to know them, they can get to know us, and uh, we can enrich the community that way.
Yeah, I really like it. And it's sort of a nice mix. Some of it is about the use of Galaxy and, learning how to get data in and how to run tools. But really the star is the science that we're all trying to take place, in my opinion, where it's like, oh, here's a research question. You're trying to get started. Often, you need to find the right data, do QC, do some sort of analysis, do some statistics, make, I don't know, various plots of it. And it really walks you through kind of end to end about how to do, these different scientific tasks. Are there any like I don't know, if you're new to the Galaxy Training Academy or maybe new to Galaxy, are there any like particular tutorials that you like really strongly recommend that people should pay attention to?
um, we have like the regular. I think the basic first we recommend is NGS data, which is you don't really need to have any knowledge in genomic. It's mostly like file manipulation, getting to know the
Yeah.
interface. Um, and once you get like a bit of a handle on that, because I know it's intimidating, it's a super powerful tool, which means there's a lot of function, and it's hard to like get yourself orientated at first. So I think this one is a good one to like just get to know where are things. But once you're a bit more familiar with interface, like everything's pretty good. Like there's a lot of debutant level training in a lot of categories.
Yeah,
So
yeah. Cool. And then I know you've been using Galaxy, I don't know, more than 10 years now. Do you have any like favorite advanced features?
the workflow got amazing. It was
Yeah.
kind of secondary when I started, like, oh yeah, you have your thing, you can kind of like do it again. But like now you have conditional, you have report, you have it gotten so much more powerful. Um
When you build out workflows, do you like to build them there's a couple ways you can do it. One is you can kind of open up the workflow editor and kind of, think ahead what tools you might need and how they're connected. Another way is to kind of run tools interactively and then extract the workflow from that. Do you have a preference on how to build out workflows?
I do by like tiny workflows. Like I have a function, okay, I'm gonna make a small workflow, test it out. And then once I have like the individual function, I kind of gather them in a bigger workflow.
Hmm.
Um, Sometimes I have doubt about like one parameter and I'm going to run it outside of the workflow, but usually I have a pretty broad idea of what's going to go together. So I go that way. Or the invocation view also is fantastic when it appeared. Made my life so much easier. We used to have only the history to go through what was the result of the analysis, and when you have workflow with dozen and dozen of output, it gets really confused really fast.
Yeah.
And so no, the people watching are gonna see when you run a workflow, you have this nice view with like every step, and you can click on a step and see what are the file.
Um, yeah.
Way easier to see the result and also like if something goes wrong, where it went wrong.
Yeah, I mean it's real it's I agree. That's an amazing feature to be able to kind of dive in. Do you have any tips or tricks, if something goes wrong, like, where should people look if there's issues and they're not getting what they expect to see? Like what are the kind of top places they should look?
Well, first step is verify that all your you don't have like a empty output. Because you have a lot of error, like the job is green, but like the file is empty, and so the next file is gonna fail, but you're not really sure why.
Yeah.
So just go back one or two steps, check that you actually have data in the file and that the file is the right format. Sometimes it might be compressed when you're expecting something uncompressed.
Uh, yeah.
These are like easy mistake to miss because they fail like one step later than where the actual problem is. Um, and usually the way I do is I when you look at the job information, you have the output and the input. Look at both, and then go to the input, look at information of the input, and kind of like go up the chain that way,
Yeah.
and check that the file are sound. Um, yeah, I think going like the three things, we catch maybe 80% of the errors. Um,
Okay. I guess another one is sometimes it just helps to retry where I don't know, cosmic ray or something weird happened and if you
yeah, if
just
you don't, yeah, that's fine. Yeah, that's true. If you don't find like a obvious error, rerunning is
sometimes that helps.
sometimes that helps, but it's fine when it's a 10-minute job, when it's a 3-hour jobs. You want to be
Yeah,
slow.
yeah. And I know one of the sort of aspirational goals of Galaxy is to make sure work is reproducible. Like, what does that mean in sort of modern data science? Like, do the files need to be bit for bit identical? What does reproducible mean to you?
Um, to me, it means being able, like, a few months later to understand what you've been doing, why you were getting the result you got. Um, because sometimes you're going to have a new version and you're going to find, oh, my results are strange. Did I do something different? Or is just like a tool version is different? So going back with what you do, rerunning it and verify, being able to like check the data size by size, and you can try to understand what, if it comes from the data, if it's just a technological differences. Um, and that's just like for the long term. For the short term, being able to, when you publish a paper, for people to be able to like reproduce and like verify the data and like see
Yeah,
what parameter you use, if they agree with the parameters you use, is super important, I think, in research. Because it's easy when you have dozen and dozen of tools to miss, to have a parameter that is maybe not the best one to use. And so being able to share it, to rerun it, and for people to have a new eyes on it is super useful.
yeah. Yeah, I mean, I guess it's both the fine details of, what was the tool and what was the version, what were the parameters. And sometimes those small details really matter, right? I've seen
Yeah.
results, flip one direction to the other. And then there's also kind of the bigger question of like trying to understand, what was the goal of this? How was the analysis performed? Making sure that it's robust. I think you want to aspire for both the fine detail and then the bigger understanding.
Yeah.
Yeah. Um, maybe now would be a good time. So I know a lot of your work recently has been centered on the Vertebrate Genomes Project. First of all, like, what is this project? What is that all about?
So it's a project, it's a global project. There's a team from all over the world working on it. Um, and it was born from desire to preserve biodiversity. And so trying to sequence, at the end of the project, every species of vertebrate in the planet, which is 70,000 or something, I think, uh, to try to preserve uh, what we know about this species, especially endangered species. And the first step in this project is getting one genome per order and then one genome per family and like go um, cover the whole tree that way.
Yeah.
Um, and there is so many people involved. There's people locally collecting the sample, there's people sequencing, and there's people assembling and curating. And I'm been working closely with the people curating and assembling to try to make the job faster and easier because the sequencing data come very fast, and so we need to be able to keep up with the speed computationally and being able to generate this 70,000 genome at the end of the project. And so that comes through finding the best balance between, oh, we want to have this high quality genome, very strong, but also we need to produce them fast.
Yeah, I mean it's really ambitious, like you said, it's trying to get all 70,000 species. You mentioned something about orders, what's the current status of the VGP?
So they just finished phase one, which is 470 or 520. I don't remember the number exactly. But yeah, I think it's one per order, plus a couple invertebrate and outside the order to kind of like balance the tree and being able to compare to make some analysis with it. So yeah, phase one is just done. There's going to be a lot of paper coming out this year and next year about both the genome, comparative analysis on it. We're preparing a paper for the workflow.
Yeah.
We had one published in 2024, which was the first release of the assembly workflow. And this new one is, we made a lot of changes to this. We improved the process a lot. And we also have additional workflow for curating because when you do assembly, the tools are getting really great, they're giving good results, but you still need to have some human intervention because there's something that the algorithm don't really see.
Mm.
And this is kind of a big bottleneck in getting this genome out quickly. And so we're working on automate making this process as automatic as possible to reduce the time spent on the manual creation. Um,
So at a high level, what does it take to sequence a genome? What do you need to do?
um, well, first you need a good sample, which is like a bit not that easy when it's an endangered species, when you can't really find the individual and you can certainly not kill them. And you need to have enough material to sequence it. So that is the first step that is difficult for some species. Then you need to ship them, which is another problem. Some sample have been stuck for months between the lab and the sequencing lab just to cross the borders. Um, yeah, and then it goes in the lab. You produce different type of data depending on what's possible, how much material you got, and what uh, yeah, what's available in the lab. And from this tiny sequence, you get raw file, and then that's where I come in, and when the people I collaborate with come in, which is okay, you have to find these like tiny puzzle pieces, find them together. It's been put in a shredder, and you just painstakingly pasting them next together. The tool does that for you, luckily. And yeah. So a lot of quality control, verifying that we don't have any mistake, that we have that the data that we have are sound and there's no like contamination, because sometimes you might have a lot of other organisms like bacteria, human people who collected the sample might have put
Yeah.
some human DNA in it. So yeah, just checking the quality over the process.
You mentioned that there's potentially different types of sequencing that might be involved. What's your favorite? What do you see as like the most important or one of the most important types of sequencing to do for this?
I don't think there's one. I think like all the data have their strengths and weaknesses, and the best results we get are when we use all of them. Uh, like
Well,
Oxford Nanopore long read, PacBio hi-fi data, and Hi-C data. Um, we supported at some point the BioNano CMAP as well. I think we come to realize it's really useful for manual curation. It's not that useful in the process of doing the automatic assembly.
yeah,
Uh, but also that might depend on the organism we're working on. We're trying to find the best balance for all the clades,
yeah.
but all genomes are very different. So
Well, you
we
mentioned,
don't know. We have this basic protocol, but
yeah, you mentioned Oxford Nanopore, PacBio, and Hi-C. Could you tell us a little bit about, what are the pros and cons of each of those three types of data?
um, so I think ONT is longer. I don't know if that's still true. They used to have more errors in the reads, but I think it got better with the new technology. I haven't worked that much with ONT yet. I know it's in the future of VGP. We're gonna start
Yeah,
integrating it. Uh, PacBio Hi-Fi had a good balance between longer read than ARIMA sequencing, short read data, and also low error rate. Um, it does have, I think, all of them, I think, have some bias in the region of the genome. I think
mm,
Hi-Fi is a bit performing a bit less in like high GC content area. So that's where nanopore might be useful if you have a species with this kind of area that are problematic. Um, I really love what ARIMA IC data allows you to do. It's a technology that allows you to have an idea of how the DNA is packed in the cell. And as a concept, I find that fascinating. I love that we can just see the sequence and say, oh, these things are like super close physically in 3D. That's really cool. And
How
so,
does that help with genome assembly? Knowing that in the cell, these DNA bits are nearby.
yeah, because like chromosome, they're like not, we used to see them in the karyotype where they're like all like neatly aligned to each other. They're just like ball of paper. Like one chromosome is going to be a ball together, and then the next chromosome is going to be a ball next to it. So if two sequences are close, they're probably like in the same ball and in the same chromosome.
Ah, okay.
So when we're not sure, oh, these things are probably uh, in the same chromosome or like close together, three, we'll be like, well, the paper, you have more chance for two pieces to be together if they're like too close in the sheet of paper instead of like being at the other end of the pages. So kind of gives you like a signal how close they're likely to be in the
Okay.
flat chromosome.
Okay. And then, okay, so you can collect these different types of data, and then you said there's some QC, but like, what about the assembler itself? Like, is there a particular assembler that you really like, or maybe what is the VGP using now?
We're using hi-fiism. So we're using hi-fiism with uh, hi-fi data for the assembly, and it uses Hi-C data for phasing. So phasing is important when we have two haplotype, which is most of vertebrates. One half of the DNA comes from the mother, another half from the father. And when you get this piece of DNA, you're not sure like what comes from one side or the other. And Hi-C, because it gives this information a bit more long distance than just the reads, tells you, oh, these two are probably coming from the same parent. You don't know if
Yeah.
it's father or mother, but it probably come from the same origin.
Yeah.
So this is included in aphaism, this version of it. Um, it also supports ONT, so we haven't done the research yet for what we will use when we integrate ONT in the process.
Okay. So
Aphaism.
so it's sort of, you have different types of data, hi-fi ASM, even if it's ONT data, you can try to do an assembly there. You get, and then use the Hi-C data for phasing and for scaffolding. But then, like, what happens next, right? So if you get, not just one genome, but maybe hundreds of genomes, like, what sort of analyses would you want to do after you have your genomes?
What is yours? Like, there's so much thing we can do. My background is in comparative genomics, so I do really love phylogenetic tree. I know they remade a phylogenetic tree based on multiple alignment and the DNA because it's not people think, oh, we know how the species are organized, it's a done deal, but it's not that done a deal, right? Like we kind of know that the tiger is probably related to a lion, but is it more closely related than to a cheetah? It's not that clear. So having more data, more good quality genome data about this species gives us better view of that. You can also see. So during my PhD, I was working on gene family and function,
Yeah.
and so it's doing a phylogenetic tree, but not at the animal level, at the gene level. So like, oh, I have this gene for blue eyes. Is that the same gene for blue eyes in the
Mm.
dogs? Maybe, maybe not. And so do they come from the same place or just gene that appears separately? And if we know this information, we can kind of see, oh, uh, this gene that cause cancer in one animal can be not that dangerous in another. And so what's the difference between the two? And it helps us learn about where the function comes from and how we can work with it. And maybe, I know people hate GMO, but I was working with, in the goal of GMO during my PhD, which was trying to find which gene were helping crop resist drought.
Ah.
And so, yeah, like this uh, type of rice is stronger in drought than the other. Why? What's the gene? And can we use this gene in other? So we avoid food shortages in drought condition. So yeah, like my first instinct is going, yeah, let's compare and align everything. But yeah, there's so much you can have aligned RNA data and look at transcription. It's a strong base. A lot of any analysis you're going to do with proteomic RNA, it's useful to have the DNA sequence to align to and to compare to
Yeah.
as a foundation. So having a strong, good quality reference, I think it's useful in so many levels.
Yeah, I guess just to kind of recap, I think, for a single species, it's a reference. So you can do functional genomics, you can annotate genes, you can try to find regulatory sequences, and then it gets really special when you have a collection. And you mentioned like traits like eye color or whatnot. You can kind of look at how the genes are evolving, regulatory sequences are evolving, try to tie it, associate it with different traits. It's really enabler. You mentioned at the end, the importance of genome quality. Like, what sort of metrics would you look at to be able to argue, oh, this is high quality or low quality? Like, what should people pay attention to?
That's discussion we keep having. The basic information is how fragmented it is.
Mm,
For a lot of species, we have some idea of how many chromosomes we should have because we've done some karyotype. So if you have as many uh, chromosome in the assembly as you expecting to be, it's probably you have a good continuity. So the
yeah.
stuff called the chromosome are whole. Um, Also, how many gaps you have, because a lot of time we know, okay, these two sequences belong to the same chromosome in that order. But this region in the middle, we don't really know what the sequence is, so it's a lot of ends. It happens a lot for centromere, for example. And so the less gaps you have, the more information you have, and that's a sign of quality as well. Um, But yeah, we've been having this discussion because we're working a lot on manual creation and how do you decide if a creation is good or not? Because we don't have the ground truth. We don't have like
Yeah.
the actual sequence to compare it to. So it's you just have one genome. Like how do you know if like it's attached correctly, if you don't have mistake? It's yeah, it's hard to know.
Hard to know. Yeah, yeah. You've been involved in the assembly now of, I don't know, hundreds of different species. Do you have any favorites?
Well, I love I like I like bad boys, so I like assemblies that misbehaved. I know there's a couple of frogs and crabs that have been nightmares to assemble because I'm working with other colleague that works on crabs as well. I know it's pretty complicated to assemble. I think we have a viper that we discovered was actually triploid and we didn't know.
What makes one genome harder or easier to assemble than another?
Um, Sometimes we don't really know. It's just like I think some species, uh, as a rule, maybe it's a way the DNA is compacted or not. The sequence, the data are hard to obtain.
Ah, yeah, okay.
Good quality read, so you have a lot of missing region, a lot of like shorter read than you're
Yeah.
expecting. Um, and you have some with a lot of repeat region. So because your reads are limited in size, if you have very long repeat region, you don't really know where to put them if it goes farther than the read. So if you have a lot of really large repeat region, it gets very confusing for the assemblers. Um, and yeah, some of them are triploid, some of them have large duplication. I I love working with vertebrate because working with plant was terrifying. Like the genome are so complicated and duplicated everywhere and messy, and that's why it's very complicated to assemble plants.
Do you have any favorite plant species?
Well, I started my career with a banana tree, so
Ah.
it will hold a place in my heart forever.
I guess at the level of, the workflows, there's a lot of parallels between plant genomics and animal genomics.
Yeah, parallels but also difference, and yeah, and it's a discussion because we're doing these workflow that we're making are great as ground, like it's going to work with the most species we can for this project. It might work for a bit more species with a couple tweak without too much work. And there are species we know you're going to have need a completely different workflow. Um, yeah.
And then you mentioned VGP is sort of wrapping up sort of phase one. What do you think will be the future of this? I mean, 70,000 genomes seems like a ton of work. Is that 70,000 years away? How long will it take to do all those genomes?
Well, we need to plot the data. I don't know. We're going way faster right now than when we started. So if we follow this curve, it should be possible. At some point, we're going to hit a computing resource problem, though,
Hmm.
because these are very intensive calculation, especially for large genome. So,
So I guess it starts with getting access to the right samples and getting through the challenges of like sequencing them. But then you say there's a sort of computational challenge as well too.
yeah, like computational financement more than it. There's probably like enough computational resource in the world to run this
Ah,
thing, but then the grants are just a certain amount of money. You can't have unlimited resources. So,
yeah,
we hope not to be a bottleneck. That's why I'm working so hard on making it as efficient as possible.
yeah.
Hopefully, we won't be an issue. But regarding the project, I don't think we'll be able to finish the project before any more species disappear, sadly. So we're probably going to
I
lose
hope
some of
so.
them in the process.
I
Yeah.
hope so. I hope so. I mean, if you fast forward, I don't know, 10 or 20 or however many years it takes, like, what are you most excited about in terms of like what we might learn from all of this? Is there any, I don't know, are there any species you're particularly excited about or traits you're excited about? You tell me. What are you most excited about to learn?
Yeah, like the phylogeny of endemic species, I'm kind of fascinated how these
Hmm.
very local species evolve, where they come from.
Yeah,
I think we're learning that with the human a lot, right? Like, oh, there's been some inbreeding with Neanderthal at some point, and we know that by looking at the DNA
yeah,
and geographic location. And I think there's so much we can learn of where the species of Madagascar come from, or some species, we have no idea how they arrived in this island, but yeah, we can learn, oh, they come from this other continent that used to be maybe close, but like millions of years
Yeah.
ago.
So it's a little bit like almost like a time machine where we can go back and kind of see the connections from millions of years ago through the
Yeah.
genome sequences. Yeah, it's kind of beautiful when you think about how we can make all those inferences. What do you think will be, I don't know, the future or maybe the role of computational biology? I mean, it sounds like it starts with finding, kind of the right samples and doing sequencing. Will the computation ever get a lot easier or will it get a lot harder? What do you think will happen there?
Um, I think both in the sense of a lot of the tasks that we're doing right now are going to become way easier, and they're going to give us freedom to find more hard things to do. And that's how science goes, right? Like once you solve a problem, it means you have harder problem to focus on, and that's the beauty of it.
Yeah, I completely agree. I mean, it used to be an enormous undertaking just to assemble one genome, but now, you've already done hundreds and hundreds. So there's sort of almost an endless list of genomes, species, questions that could be asked from it. So that's the nature of the field is to keep advancing. I have seen there's a lot of interest and excitement around like AI technology. How do you feel about that? Is that something you're excited about to help with this, or are you, I don't know, have concerns about all the hallucinations and mistakes it can make?
Yeah, I think the hype is high, but it's a dangerous path to take because you don't know what it's doing.
Yeah,
And when you can verify, like for coding and developing tool is great because you can actually verify. You have the ground truth that you're expecting,
yeah,
so you can check. For analyzing data, you don't know the truth, so you can't verify what it's doing. And I think it's very dangerous to trust the AI with any kind of decision making. Um, and even if it was not hallucinating, which I don't think we can avoid it doing it, AI is made for a purpose, and it's going to be very good at this purpose, but it's not necessarily the right goal in every circumstance.
Hmm.
Like if you create an AI to, okay, I want to be super efficient at, I don't know, detecting cancer, like breast cancer or whatever, and it's going to
Yeah,
be, yeah, very good at detecting it, and it's going to detect it too much. You know
no,
what I mean? Like it's always what the metric of success are that are going to be hard to define, and uh, you can have a moral AI. It doesn't exist. Like if you have an AI who say, "Oh, the goal is to save money for the company," it's going to do it whether it's a good way or a bad way because it's doing its
hmm,
job very well.
yeah.
But I think you need this level of human. I don't like morality because it's kind of preachy, but yeah, you need some like safe gate, I think, of a human decision between the AI and anything you do.
Well, you mentioned it like it's good for programming and it's good for certain tasks where you can really, look at it carefully. So I guess that's sort of maybe the takeaway is, even though it could be doing things, you really have to understand it. You want to be able to look at it, interpret it, make sure that it's sort of, doing the appropriate analyses there. So I think that's a place where Galaxy really shines, where you can have these like really complicated workflows, but you can go back and inspect them and look at them. So I like to think, even if the way we interact with Galaxy may radically change, right? It could be a lot of, natural language, just chatting with a chat box. It's really valuable to be able to kind of peek under the hood and really be able to inspect all the
Yeah,
work going on.
it's great for advice when you don't know what to do. It's gonna
Yeah.
guide you. Uh, but yeah, you still need to be able to understand if you want to get good quality results at least. Like for exploration is great. Let's try thing everywhere. Let's install thing everywhere is great. Trying it out, but um,
Okay. And then really just my last question, on the Galaxy Training Academy this week, we have a lot of experts. We also have a lot of newcomers. Do you have any kind of final thoughts or tips, especially for the newcomers and they're not really sure how to get started or what to do? What would you recommend for them?
um, I would recommend to interact with us. Like we are a great community and at all levels. And if there's any issue or thing you don't understand, or even something like, hey, I would love to be able to do that. Maybe we can, maybe we cannot, but we want to hear about it. And that's how we progress. And I think uh, Galaxy is a great tool. It allows to do a lot of things. But for me, what I love the most about it is really like the whole community around it, people who
Yeah.
are like passionate about it, involved in it, and always happy to teach and learn and collaborate. And I think that's a great work environment to do good science and to love your job because I think it's important to have a good time as well.
Yeah, I really like that. And if people need help this week during the training academy, what's the best place that they should go?
So you can contact us on Slack. That's the best way. We're gonna have people all around the clock on it. At most an hour, maybe in transition when people are not awake yet and other people have left already. But yeah. And even beyond the training, if you keep working on it because you want to explore more or need more time because you are busy that week, we're still on Slack, not just as connected, so it might take a little bit longer to reply. But yeah, don't hesitate if you either don't understand something in the training or like are stuck, or even if you want to learn more, hey, I have this data now. I see
Yeah.
you doing that. Like, what do you guys think? Is it a good idea to try to do that? And we can discuss it, and it's interesting.
Yeah, I really like that. And that's something I really love about Galaxy because it is a global community, right? Someone in the world will be up any time of the day and will be happy to help you and chat with you. I guess in addition to the Slack, I'd also highlight, we have this Matrix channels. That's another good forum. We have, help boards. Yeah, we're here to help you. We're here to support you. Okay, well, Delphine, thank you so much for your time today.
Thank
Also, good
you.
luck to everyone at home with the academy. And then feel free to reach out to us anytime to help you through all the exercises this week. Thanks,
Good
everybody.
luck, everyone. Bye
All right,
bye.
thank you so much. Bye now.